All right, good morning, everyone. So welcome to the second class of expression of recombinant proteins, expression in eukaryotic cells. So as usual, we will start with a recap of our last lesson on Tuesday. So just to review, or in case any of you missed the class, so what we talked about was protein expression in bacteria, and we talked about different advantages and disadvantages of bacteria. So in particular, of course, a huge advantage is that it grows very, very quickly. So if you are trying to express a lot of protein, this is probably one of the easiest and fastest way to do it. It is also cheap. Definitely whenever you're working with a smaller organism, it is cheaper. But some of the challenges with working with bacteria is that the codon usage can be a bit different, especially if you're trying to express a mammalian protein. There's also some challenges when it comes to post-translational modification, and we'll talk a little bit about what those are and how eukaryotic systems will get to address that. And also with bacteria, depending on what strain you use and what plasmids you use, you have different copy numbers, and that also affects the quality of the protein that you're expressing. We also talked a little bit about the essential genetic elements for the expression, particularly the promoters. So hopefully you can remember what were some of the host versus phage promoters and how they differ. We talked about how RBS, ribosome binding site, is important, different stop codons, and of course replication of origin. So again, replication of origin does affect the copy number. So hopefully when you hear things like PUC or PBR332, it automatically comes to mind whether those are low or high copy number plasmids. We also talked about different cloning strategies, such as how you could use restriction enzyme or fusion proteins to direct your protein where to go, or to actually introduce different types of genetic elements into your recombinant protein. Lastly, we talked about an overview of available expression systems and expression strains. So again, the name does tell you a lot about how the plasmids work or how the strains work. So for example, if you see something that has PLAC in it, you know that you need a lactose as a source in order for you to successfully express your protein. All right, so this is still just a recap. So when we talk about protein expression systems, again, they can be intracellular or extracellular, and they can be directed out of the cell if you add a signal sequence or a fusion sequence. It can also be a homologous or a heterologous production. So homologous means it comes from the same organism, and heterologous means you're actually getting it from a different source. And of course, you can produce it in autonomously replicating plasmids. Sorry, my computer is a bit laggy today. All right, so why would we need to actually express it in eukaryotes then? So with prokaryotes, when you are expressing protein or recombinant protein, you do have a couple of challenges, as we pointed out earlier. So when we do look at eukaryotic protein, they do have certain advantages. So that is why we will be looking at eukaryotic systems today. So today's overview is we will be going over different protein production steps. We'll talk about how you can express protein in yeast systems. We'll talk about two different yeast systems, bacillovirus insect cell expression, mammalian cell systems, and something that would be good to remember or your take home from today's lecture would be what are the differences and the advantages and disadvantages of each of the different systems. And then we'll end with the workflow of the cloning, expression, and purification of recombinant protein. All right, so when we talk about protein production steps, it can be divided into four main components. So the first one, which we saw quite a lot about in the last class, is how do you actually express your gene in a new host? So how do you regulate the expression? So at least in prokaryotic systems or in a bacterial system, we saw that things like your promoter actually affects it. We also saw the type of plasmid and the type of strain that you use also affects it. We also touched a little bit on molecular biology methods. So this does sort of go back to your cloning scheme, for example. So that's one of the first important steps. Next is the post-translational modification. So we did learn that in bacterial systems, bacteria is not exceptional at post-translational modification, which does mean that you may get a slightly different protein structure or protein function from its initial purpose. So when we talk about post-translational modification, certain portions that are important to think about is how does it affect your folding in any modification, how does it affect translocation, and how does it affect glycosylation. Next is protein secretion and intracellular transportation. So again, where do you actually want your protein expressed? And is your system actually doing that? And then finally, the last step is your downstream processing. And in this case, you're talking about your protein engineering itself and your recovery and purification. So these are some of the genetic elements of your protein production in eukaryotes. Hopefully when you see this, you can sort of do a very quick comparison of what it looked like in prokaryotes. So you do see similar portions. So for example, your promoter is also seen in eukaryotes, although your eukaryotic promoter is different from your bacterial promoter. You also have a signal sequence. So this is what actually directs your protein production out of the cell. You also have introns. So this is found only in eukaryotes. Although when you talk about cDNA, which we talked a lot about when we first talked about what you would actually use to produce your protein, the cDNA does not contain introns. And of course, as what we saw in bacteria, different organisms have different codon preference. So for example, if you're trying to code for proline, a proline in yeast would be very different from a proline in bacteria. So we talked a lot about post-translational modification in the past, and I think you guys might have learned this in some of your other classes, but just a quick recap when we talk about post-translational modification, basically all of this sort of falls under that. So whether it's phosphorylated, whether it's glycosylated, your ubiquitination, acetylation, methylation, though these are more epigenetic sort of modifications. But they are very, very important. Glycosylation in particular is species specific. And we will sort of talk a little bit more about that a bit later. In particular, when we talk about glycosylation, you have N-glycosylation, which occurs in asparagine, or O-glycosylation, which starts with serine or threonine. And again, we'll go over what those mean a bit later. You also have many chaperones, which help with protein folding. And again, if you have your protein sequence, but it doesn't fold well, you may not get the structure or the function that you want. The signal sequence that we saw earlier that came just a little bit after your promoter, that's actually cut off before the protein leaves the cell. So that actually doesn't affect the protein structure and form at the end of it. All right, so those are the basics of protein production steps. A lot of it should be almost like a review because we did see a bit of it when we were talking about the bacterial expression systems. So now we'll move on to yeast systems. So one of the most common yeast that's used in recombinant protein expression is Saccharomyces cerevisiae. And if you look at table 7.1 here on the right, there actually has been lots of recombinant proteins produced by the cerevisiae expression system, including different vaccine antigens and proteins, even for diagnostic purposes and therapeutic agents, for example, epidermal growth factor and human growth factor that someone pointed out that they were interested in producing as a recombinant protein. So the reason why it actually has been used so globally is because it actually is something that is extremely well characterized. So it's also small, so you can grow it up quite easily. Something that's really good about Saccharomyces cerevisiae, it has a very strong promoter, so it is very easy to regulate the amounts of protein that you want. Something that is definitely an advantage over bacterial systems is that it does carry out post-translational modifications, and mostly is recognized as safe by USDA and FDA. So as long as it's safe, you know that you can actually use it to produce other types of proteins. All right, so in particular, there actually are lots of different yeast vectors out there. We will be going over primarily three today, but the four main classes are YEAP. So the names actually do tell you sort of what the function is. So YEAP actually stands for yeast integrating plasmid, and it's integrating plasmid because it actually helps with integration into the genomic locus. So you actually see the image here on the right. There's also YEP, which is yeast episomal plasmid. It actually is able to replicate multi-copies of the plasmid, and it's primarily used for recombinant protein studies or multi-copy suppression studies. You also have YCP, which is yeast centromeric plasmid. That's a replicating single copy plasmid. So again, that's how it differs from YEP. So YEP is a multi-copy, whereas YCP is single copy. And YCP is primarily used for complementation studies, and you also have YAC, yeast artificial chromosome. If you do look across all the different yeast plasmids, you do see certain similarities. So you do see, for example, they all have a selectable yeast marker. And this is something that you will see across any yeast work that you will possibly do. You do see that some of it has a bacterial replication origin, but some of it don't. Some of it has sort of unique features. For example, with YEP, you see that is a 2-micron plasmid DNA. I'll explain what that means a bit later. And then with YAC, which is the largest out of all these plasmids, it even has things like telomeres, which sort of differentiates it from all the other yeast plasmids. All right, so let's quickly dive into YIP in detail. So YIP is, so most heterologous genes are provided with a DNA coding sequence for signal peptides that facilitate secretion. So again, this is sort of generic. Whenever you see any signal peptide, it is usually for the purpose of helping with secretion of the protein. These amino acids are then equipped with a protease cleavage site so that they can be removed from the recombinant protein. So again, this is mainly so that at the end, your final product is your specific recombinant protein. But what's useful about YIP is that it's used to integrate a heterologous gene into the host genome. It doesn't carry an origin of replication in comparison to some of the other yeast vectors that we saw earlier, but one of the disadvantages, it has a low yield of recombinant protein because it only comes from a single gene copy. So if you look at the image on the right, it's the diagram version of everything that I just explained earlier. And how the integration often works is usually whenever you hear anything related to integration, you often have homologous sequence, which actually enables homologous recombination to occur, which is what you see here, the red regions. So anything that's sort of in between the homologous regions, that is what can be transferred to your chromosome of interest or actually help you with the integration into your chromosome of interest. So in this case, they were trying to introduce both the gene of interest and a selectable marker, leucine-2, which is typically found, LU2, sorry, which is typically found in yeast and is typically used as a selection marker. So a little bit about YAP now instead. So hopefully after I sort of talk about this, you can also sort of see the difference between YAP and YIP. So YAP, again, sort of the distinct feature is that it's a high copy number 2-micron plasmid. It does replicate independently through a single origin of replication. So again, something to remember is that not all yeast vectors have an origin of replication. But because this one does, and because it is a relatively high copy number plasmid, there's more than 30 copies per cell. So automatically, if you sort of compare between YIP and YAP, you know that YAP typically would produce more protein in comparison to YIP. Um, it does rely a lot on certain mutant host strains. So, for example, histidine, tryptophan, and leucine. There is a diagram a bit later in one of the later slides that will actually show you how this works. So, don't worry too much about it if you can't quite imagine how the selection markers are used. But basically, to sort of simplify it, when you have a YAP with a wild type LEU2 gene, and it's transformed into a mutant LEU2 host cell, only cells that carry the plasmid will grow. Again, there's a diagram later on, so that will probably make it a bit more clearer. It is highly regulated, although inducible promoters are preferred. So inducible promoters basically mean that you can turn it on or off depending on when you want your protein to be expressed. So this is just a very sort of simplified table about what genes you have and what proteins they can produce, and some of the different inductions that you can use. So for example, when you see something like PGK and GAPDH, often you have a carbon source and you'll be able to express those. We move on now to YAC cloning system before moving on to a different yeast cloning system. So YAC is easily the largest out of the 4 different yeast cloning systems that we have seen so far. And since it is large, it actually is able to accommodate or clone a very large segment of DNA, roughly about 100 kbs, which is huge. And it's then actually maintained as a separate chromosome in the host yeast cell. So especially if any of you took my class last year, or last semester, something you can think about is what are the advantages and disadvantages of having a chromosome maintained separately. If you didn't have me last semester, something to think about as well is, can you think of reasons why it might be good to maintain a chromosome separately as opposed to integrating it using the YIP system? So with YAC, it is highly stable, and because it is so huge, it actually has been used for mapping of human genomic DNA. So this is primarily because when you are trying to clone a gene or to be expressed as a protein, if your system can only accommodate, say, 1 kb and your gene is the size of 5 kb, then obviously you wouldn't be able to express that in the first place. So YAC is definitely fantastic for that. And it's actually also being used quite a lot for fundamental studies to analyze transcription units and even genomic libraries. It has sequences that can act as an ARS for replication, centromere for cell division, and telomere for stability. So this was also pointed out earlier. If you sort of remember the linear genomic image or genomic map of YAC, you did see telomeres sort of at the 5' and 3' end of the YAC cloning system. But what is interesting is that they have actually not been used as an expression system for commercial production. So again, even though there are definitely lots of advantages and benefits to YAC in comparison to YIP and YAP and YCP, it actually hasn't been used as an expression system and has been mainly used more to actually study different physical genomic units. All right, so when we talk about yeast gene technology, often something that's sort of really important is how do you actually do transformations? How do you actually introduce certain genes into the yeast? The main steps or the main methods are through spirosphere transformation, whole cell transformation using lithium acetate, and electroporation. So just to quickly walk you through what's on the images here on the right, this is how spiroplast transformation occurs, the image in white here. So you get vegetative cells, and then when you have a cell wall lytic enzyme, you actually get formation of spiroplasts. And then you can actually add your plasmid DNA together with PEG or calcium. So this is typically used in all transformation methods, so not just yeast specific. And then that actually enables transformation to occur. Overall, this whole process takes about 1 week, which is not too bad. With certain other organisms, the transformation time is a bit different depending on the organism. This one is sort of utilizing a selection marker. So you have competent yeast cells lacking URA3 gene, And then if you incubate it with plasmids that has URA3 gene, it's then able to take it up, and then your final cell would actually contain URA3. So you can actually then select for it with medium that lacks uracil. These are some of the other methods. So electroporation is pretty straightforward. You actually have your yeast cells together with your DNA of interest. Electroporation is pretty much like an electrical pulse zap to the cells, which actually changes the cell walls and then enables it to uptake it. And then again, you can use a selection such as your tree or leucine to actually pick out the cells which now express your protein of interest. With lithium acetate, it's your yeast, and then you add lithium acetate to it, and then you add PEG, your DNA, and your plasmid DNA. You heat shock it. So these are all, again, common methods, whether it's electroporation or heat shock, and then you plate it with a plate with a special selection media as well. PEG is you have your enzyme with your yeast, which is then converted into protoplast. You add PEG again together with your plasmid or your linear DNA, and then goes through a selection plate. We won't really talk about agrobacteria too much, but this is alternatively a different method of actually doing transformation. So just to sort of quickly point out certain information, your transformation can be done through complementation of oxotrophic mutations. So again, this is sort of what we see in this diagram here, where your strain carries a particular mutation, and what you are trying to introduce contains the wild copy of the gene. So when you do that, then your final product will actually have the wild type copy of the gene, and then you're able to select for that. You also can select using other dominant selection markers. So for example, if a particular strain is resistant to G418 or formaldehyde or cycloheximide, that can also be used as a selection to identify which of your cells now contain your gene of interest or your protein of interest. Okay, I'm going to quickly stop here. Any questions so far? Okay, no questions? Okay, so I think I'm going to continue. It looks like there's no questions. Right, so next we move on to secretion of heterologous proteins or PTM. So as pointed out a couple of times earlier today, proteins may also be produced for secretion. And in this case, any glycosylated protein is actually secreted, whether it's O or N linked. How it works is you have the coding sequence of the recombinant protein has to be cloned downstream of the leader sequence. So that's the L in red. You see it here and you see it here. So if there is proper disulfide bond formation, you actually get removal of the leader sequence. So you actually see your A being bonded to your B, and then everything else is cleaved out. And what you have left is your active recombinant protein, which is secreted. So again, what's sort of important is you actually have your leader sequence here and proper disulfide bond formation. So how does post-translational modification actually work? We've heard many, many times how it actually is very crucial. And this slide actually summarizes it quite well. So glycosylation happens because it's a reaction in where a carbohydrate or a glycosyl donor is attached to a hydroxyl or another functional group. So in this case, you have your N-linked or your O-linked glycosylation. It is very important because it actually does help determine your protein structure, folding, function, and stability. So if anything is changed, again, even if you have the exact amino acid sequence, but you don't have proper glycosylation or structure formation, you end up with something completely different. So the most common glycosylation is when you have attachment of specific sugars to hydroxyl group of either serine or threonine. That's for your O-linked glycosylation at the bottom here. So the sugar groups that I mean can include mannose, galactose, fucose, basically any of the sugar sources that linked or that's listed down here. You can also get sugars linked to amide groups of asparagine. So whenever you have asparagine as sort of the base, that's an N-linked glycosylation. So it's quite easy to remember. If it's asparagine, it's N-linked. If it's serine it's O-linked. All N-linked glycosylation in eukaryotes start with the same initial group, which is subsequently trimmed and then elaborated in different ways among the species. So you do see that it can be quite diverse, especially in N-linked glycosylation. That's why they sort of have even different groups for it, complex, hybrid, and high mannose, whereas in the O-linked glycosylation, it's a bit more simpler. So we talked a lot about Saccharomyces cerevisiae, but there are sort of, again, pros and cons to every single system. So with Saccharomyces cerevisiae, the downside is that the protein production level can sometimes be quite low. Even though PTM, post-translational modification, is relatively good in Saccharomyces cerevisiae, sometimes you actually end up getting hypoglycosylation, which still also results in the change of protein function. Sometimes the proteins are also not delivered correctly to where it's supposed to go, again, depending on the signal sequence. But when it is retained in periplasm, that actually increases time and cost for purification. If you remember from the first lecture with me, one of sort of the end goals or the end step for protein production is you do need to produce a large level or a high level of protein, which is typically done in a bioreactor. And at that stage, it actually produces ethanol at high cell densities, which is toxic to cells. So there really sort of needs to be a good balance. So to sort of combat that, some people use Pichia pastoris, a different type of yeast that is actually able to utilize methanol as a source of carbon and energy. Glycosylation occurs to a lesser extent in Pichia pastoris. And it's actually, this is a strain that has actually been engineered in such a way that it's actually capable of developing a more humanized strain. So the upside of that is when it's more humanized, then it is quite similar or more similar to a human cell, which means it's something that's sort of easier to work with, especially if you're trying to express a recombinant protein. One of the plus points is definitely that it doesn't produce ethanol and it secretes very few proteins, thus simplifying the purification of secreted recombinant proteins. Right. Whew. All right, we are done with yeast systems. I'll pause again to see if there are any questions. If not, I'll continue. Today's lecture is a bit long. Right, I'm going to continue then. So next we move on to expression of proteins in baculovirus insect cell expression. So baculovirus is a large, diverse group of virus that specifically infects arthropods and is not infectious to other animals. During infection, there are two forms of baculovirus that is produced. So if you look at the image here on the right, you actually have your single nucleocapsid, which is this sort of long thing here. It's a virus particle which can infect mid-gut cells. You also get a cluster of nucleocapsids that are produced outside of cells in a protein matrix. This is known as polyhedrin. So it's this sort of hexagonic shape that you see in B. The polyhedrin is actually quite important. You will see later on how the polyhedrin is what is replaced with coding sequence for your gene of interest or your protein of interest, and that is what actually enables production of heterologous protein. So with baculovirus insect cell expression, if you look at the table here, again, tons of recombinant proteins have been made using this. You see a couple of antigens, including dengue virus. Antigens have been produced with this, HIV, influenza virus, IL-2, malaria proteins, and other types of antibodies and glycoproteins. Most of these proteins are modified and secreted properly, and it grows very well. But it does not process certain mammalian glycosylation types correctly, such as galactose and anything that is N-linked. So the bacillovirus vector that has been used extensively is known as Autographa californica, multiple nuclear polyhedrosis virus, such a mouthful. So the short form of it is known as ACMNPV, which is what you see down here and also what you see in the image here on the right. So how it works is you can actually insert your gene of interest into a multiple cloning site, and then the transfer vector is propagated in E. coli. Next, your insect cell in culture is co-transfected with ACMNPV DNA and your transfer vector. So if you look at the image on the right here, again, you have your ACMNPV genome and your transfer vector. You do have homologous regions between both your transfer vector and your ACMNPV genome. Again, remember your polyhedrin is what is usually swapped out for your gene of interest. So in this case, since your gene of interest is on your transfer vector, after homologous recombination occurs, your gene of interest then is transferred into your ACMNPV genome. And now this recombinant plasmid or genome actually contains your gene of interest. So the ACMNPV DNA can be maintained in the plasmid with kanamycin resistance gene as a selectable marker. So we do see this sort of over and over again. And they carry backmid, which is co-transformed with a transfer vector and a helper plasmid. So I know that's a lot, but I'll explain what that means in a bit. Finally, your bacteria with recombinant plasmids can produce white colonies in the presence of IPTG and XGAL. Because it carries a kanamycin-resistant gene, the colonies are resistant to kanamycin. And then your final recombinant backmid can be used to transfect into insect cells. So again, you start off with your ACMNPV genome containing a polyhedrin gene, and then your transfer plasmid. In this case, your homologous regions are here, the 5' and 3' region. So after homologous recombination, you get this entire region, which is now in the ACMNPV genome, or your backmid. And then you have a different transfer vector. Again, you see the homologous regions. These are where it's in green, and together with the helper plasmid. So what ends up happening is then you're able to get all of this, your kanamycin resistant gene and your gene of interest to be transferred here in between your LAC-Z areas. So it is pretty similar to what we saw in the slide before. It's just that this one has one extra step. All right, any questions so far? Otherwise, we'll move on to mammalian cell system and workflow of cloning expression. No question, right? Cool. I'll move on. Hopefully we can finish in time. All right, so next we move on to mammalian cell expression system. This is definitely the best when you are trying to produce protein with post-translational modification. Many established cell lines have been very useful with mammalian cell expression systems. For example, human embryonic kidney, Chinese hamster ovary cells, and they can be either expressed short-term, transiently, or for a long-term expression. The expression vectors in the systems are derived from animal virus, such as SV40, and can be used for expression of single polypeptide, homo-oligomers, or heterooligomers. But mammalian cell expression systems can be pretty pricey. So this is what a typical mammalian expression vector looks like. Again, you have your promoter. You also have things like polyadenylation and your terminal sequence. Your gene of interest should also have a translational control sequence, and this helps facilitate secretion and purification. There should also be a Kozak sequence, so that's usually at pretty close to the start codon. You have your signal sequence, you have a protein affinity tag for purification, a cleavage site, and a stop codon. You also have a 5' and a 3' UTR, and that increases efficiency of translation and contribute to mRNA stability. So when you look at all these different vectors, hopefully by now you are able to tell what the different components are, what the different genetic elements are, and which is found in which vector. Right, with mammalian expression systems as well, you also have what is known as the two-vector expression systems. And even though you see three different vectors or expression systems here, they all give you the final product, which is similar, which is an assembled protein of alpha and beta. So I'll quickly walk you through what all the differences are. So in this case, you can get a single plasmid expressing gene alpha and another one expressing gene beta. In both these cases, you can imagine that they produce separate mRNA, which are protein subunits, which can then be assembled together. So this is definitely one way of doing it, but people try to not use this so much because you're actually utilizing two different plasmids of two different systems. The one in the middle also is used quite frequently. So this is where you have a single system and you have both gene alpha and gene beta being expressed on it, usually under a single promoter. So you actually end up with, again, alpha and beta subunits, which are then assembled together. But the challenge with this particular system, the one in the middle, is that with certain expression systems, whichever which is expressed closer to the promoter is expressed at higher amounts. So then you might actually get more gene alpha in comparison to gene beta. So the one on the most right is the one that most people prefer when they have a two-vector expression system. So in this case, you have something like, again, gene alpha, gene beta, but under a single promoter as well. But in this case, it is separated by iris. And how iris actually works is it actually does cleave off eventually, but it's not as large as a promoter, but it does sort of function like a promoter. So then you actually get both alpha and beta being expressed sort of under a different promoter. So they're expressed at same amounts, and you do get an assembled protein of alpha and beta at the end. So again, by preference, the one on the most right is the one that's preferred, but the one on the most left is also okay. So we've talked a lot about different selectable markers for mammalian expression systems and all different expression systems. And in this case, you do see some that are quite similar with yeast, for example, like G418, but you also have things like bleomycin or blastocidin or puromycin. So if you're trying to sort of compare this to what you've learned in bacterial systems, it's quite similar to the function of, say, kanamycin or ampicillin. So we also have different strategies to increase yields of recombinant mammalian cells. In some cases, stress actually does change the amount of yield in certain cells. So I'll sort of just focus on this one here. If you have cells which are unstressed, you actually don't get the transcription factor being produced. But whether it's stressed or engineered, you actually get different amounts of your transcription factor being produced. It really sort of depends on what you're trying to express and how much stress you're applying. So when I talk about stress in this case, even things like heat shock or things like electroporation, which is an electrical pulse, all also counts as stress. Another strategy to increase yield of recombinant mammalian cells is through the chromatin structure. So hopefully, again, you remember this from some of your earlier classes. But if you want to have enhanced expression and stability, if your target gene should be integrated into euchromatin, so this on the left here, which enables active transcription rather than heterochromatin, because if it's tightly wound up like this, you don't get as much transcription. So the techniques to relax your chromatin structure, so to have a euchromatin structure as opposed to a heterochromatin structure, is to increase the expression of genes, including modifying host strains to express proteins that alter chromatin structure. So again, if you can have your cells or your genes being expressed under euchromatin, you actually get more transcription in comparison to heterochromatin. All right, we are down to the last part, workflow of cloning, expression, and purification of recombinant protein. I'll finish this part and then open up for questions. Oops. I'm going to skip this for today since we only have a couple of minutes left. So desired properties for protein production in hosts, we've sort of seen this throughout all the different expression systems that we have learned about today. But you do need the type of vector strongly influences the replication and type of integration. So for example, if you have something like YIP, obviously that allows for integration, but if you have something like YEP, that only allows for episomal expression. The selective markers also make a difference. So if you have something where you are able to introduce wild type leucine 2, then you are able to quickly select for something efficient transformation. So for example, if you have electroporation available in your lab, that works better than say lithium acetate. Your protein production is also influenced by things like your promoter. We've seen lots of different types of promoters, and obviously some are able to give you more protein compared to the others. Codon usage as well. If you are trying to express mammalian protein in a bacterial or a prokaryotic system, obviously the codon usage would be a bit different. Post-translational processing or modification is very different across all the different systems. So again, depending on what you're trying to express and the structure and function of it, you should select a different expression system. Product stability, this is sort of related to number 3, and GRAS status, so is something safe or not for protein production. What limits protein overexpression? So you also have microbiological strains that affects it. So when we talked a little bit about BL21 and the different variations of BL21, for example, that definitely affects protein production, gene usage, and induction. We saw that different plasmids, different strains need to be induced differently. mRNA stability, translational capacity, growth rate, protein folding, secretion, and degradation. Chemical and physical can also affect protein expression. So chemical, again, are you using a chemical inducer, or physical if you are using something like temperature? So again, if you are using a promoter such as H lambda's promoter, temperature actually affects the growth rate. Very quick overview of protein expression system. I'll just go over this briefly. You can go back and read this in detail later. So we talked a little bit about bacteria, yeast, insect, and mammalian expression systems. They do have different common applications. For example, bacteria and yeast are used for structural analysis, but for antibody generation and mammalian systems, people do use it primarily for antibody generation, protein, functional assays, and protein interactions. There are different advantages across the different systems. Hopefully you are able to remember some of them, but things like if you are looking for high levels of PTM, then mammalian cells would be the best. There are also challenges as well. Protein solubility is one of the largest challenges when it comes to bacterial systems. Very brief summary again of the comparison of expression hosts from low to high. So if you are looking at speed, highest is definitely bacteria. If you're looking at cost, lowest is definitely bacteria. If you are looking at yield, yeast is actually one of the best. For PTM, mammalian is the best. And for FDA approved, insect cells is the lowest. Comparison of expression systems. We talked about this across all the different systems. You can just go and look at it later, or you can also just sit down and try to put this together on your own and then just compare this to the chart. So looking at things like cell growth, expression levels, protein folding, and glycosylation. Workflow of cloning, expression, and purification of recombinant proteins. Almost towards the end, I think we just have 4 or 5 more slides to go. So we saw this sort of in the last class as well. You start with your gene, you clone it into your expression vector, you transform it, you grow it into your organism or expression system of interest, you grow it up, you purify it, and then you finally have your final product. So protein purification can be done through different ways, and it can be separated based on size, charge, or specific binding. So if you're talking about column chromatography for purification, you can actually use chromatography where different proteins interact with different matrix to different extents and flow at different rates. So depending on the rate or the fraction, you are able to purify your protein of interest. You can also look at gel filtration chromatography. This is when it separates according to size. So how it works is larger proteins are excluded from the pores and therefore they flow out quite rapidly. Smaller protein enter the pores and flow slowly. So depending on when you collect your fractions, you can have larger or smaller proteins. Ion exchange chromatography, it's separated by charge. So your beads have different charge groups. Positive charge binds to acidic amino acids, negative charge binds to basic amino acids. So again, because of that, you are able to sort out or get your different proteins based on affinity, or you can also elute at different salt concentrations. Affinity chromatography is separation by specific binding. This is usually when you have your proteins tagged, for example, to a GSD tag, and then you can elute out based on the tag. And finally, you can separate and visualize proteins using SDS page. So in this case, if you use an SDS page gel, you actually get the first step where you denature your protein and then reduce your disulfide bonds and then finally migrate to the gel matrix in electrical field. So your final visualization looks like this on the right, where you can actually stain your gel and check to make sure that your protein or your recombinant protein of interest is actually the size that you expect it to. If it is folded wrongly, or if there is something wrong in the production of the protein, it will not be the size that you are expecting. All right, so this is the last slide. So just some things to think about at the end of today's class is how do you make a recombinant protein? This is quite an important question. So are you able to sort of walk through all the different steps? What are the differences between each expression system? This is an important question. Are there similar genetic elements between each expression system? So is it only promoter or is it something else? What is the main criteria used to produce a large recombinant protein? So we saw lots of different things from the size of the plasmid to whether it integrates or not to how you can purify it. Which do you think is the main important criteria? How do you verify that your recombinant protein was folded properly? So do you think it's something that you sort of look at early on when you're cloning, or is it something that you can only do when you're trying to purify and isolate your protein? Can you use techniques such as PCR to verify the size of your recombinant protein, or are there other ways to do it? And what strategies would you use to increase the size of your protein? All right, so I'm quickly going to end here, and I will take any questions if there are any. I suspect you guys have class after this. Couple more minutes. So yeah, open for questions. Otherwise, as usual, you can email me, WhatsApp me, anything at all, and anything that I think is applicable to everyone, I will send out through the group. Otherwise, if there are no questions, please remember to fill out the attendance form. And if there's nothing else, I'll see you guys next week. Okay, I see some thumbs up. So I think that's it. All right then. Thank you everyone, and I will see you guys next week.